Okay, so good evening. Welcome to today's class. Just a quick recap that this we are going to have the 21st class, I think, for this course. So we will have four more classes. So in the next three classes, I'll cover whatever remaining topics there are. In the last class, I'll keep it open for discussion and also maybe just a quick recap, kind of a simple MCQ test of all the remaining topics. So don't stress too much about it. It will anyway be the last class. And if there's anything that you wish to discuss, anything you want me to clarify, some topic that is personally relevant for your own work or your research career, or if you're a student, whatever you're pursuing. So in that direction, if there's anything that lies within the scope of that course, you can let me know so that I'll prepare that material in the meantime in the next 1.5 weeks or so, and that can be the last discussion for our for this course. However, today what I have decided that I'll cover today is what we had initially discussed in the week before that we would start chromatin accessibility profiling. So, before we get into the details of that, I wanted to quickly revise the last class's discussion, which was on ChIP-seq before we went for the test. So, if you remember, in ChIP-seq data analysis, we had talked about the fact that first we need to learn how to retrieve ChIP-seq data. And for retrieval, I had shown you two possible resources that you can use. Most commonly, what we normally use is called the geodata set, which is Gene Expression Omnibus, and there you can feed your GSE ID, which is the ID of the data set, which is publicly available and which is provided by the author, so that anybody who wishes to access that data for their own analysis can do so. So if you have access to that data, you can directly put in that, or you can put your keywords which are relevant to the hypothesis that you wish to explore. And once you have done so, you'll get a lot of hits. You can filter the hits according to your requirement. And among them, then you can opt for the study that is most relevant to your analysis. And then you'll find the list of all these samples. From there, you can pick whichever sample is relevant for your discussion, for your analysis and for subsequent discussion. But what you should always keep in mind is that apart from your treatment conditions, you should always have the input. And also, preferably, if possible, you should also opt for studies where they have replicates for whichever analysis you are looking forward to. Like, for example, here, I don't see replicates, but for example, if we had treated conditions, preferably we should have two replicates of the treated conditions. However, because it's expensive, it requires a lot of resources. The amount of antibody that goes into each chip experiment is also a lot. So it's not always that people usually opt for replicates in the sequencing process, but it is advisable. And also in today's time, it has become a little more preferable. This was a study published in 2013. So back then, stringency regarding these things was a lot less. So people could get by with a single replicate data. But if you are trying to analyze something and you can get replicates, always opt for it. Then we talked about the relevance of the input file, and in context of ChIP-seq data analysis, input file, as we have discussed before, is extremely crucial because for quantification and to make sense of your data, the input file is absolutely necessary, because relative to that, you will get the comparison of your experimental condition. So the input simply is your complete cell lysate, which does not have any antibody pulling anything down. So it represents the physiological condition of the cell at the time that you have scraped it. So the only variation between that input sample and your sample of interest is the fact that there is some antibody which has been used to pull down your protein of interest, or has been used to pull down any non-specific binding using IgG. So if you do not remember, usually for any chip experiment, we have three conditions. One is input, second is IgG, the isotype control for the antibody that you're using. Why is that important? That's important because whenever you're using an antibody to pull down a protein of your interest, it might so happen that the antibody's FC region non-specifically binds to some XYZ random protein within the cell. And when you run a Western blot, it can give you incorrect results and incorrect interpretations regarding some proteins binding. So to make sure that you can remove that noise or that background, you should always have an isotype control. And apart from that, what you have is your protein of interest that you pull down. So these were the three conditions that need to be taken care of whenever you're performing a experiment. Now, after you have completed that and you have eluted the DNA, which has gotten pulled down with your protein of interest, and you have sent it for sequencing, what you get are your sequencing reads, which are FASTQ files, raw FASTQ files that you get as output from your experiment. So you can use that. And these are the steps that need to be followed when you are performing analysis. First, you need to do pre-processing or quality control, which, if you remember, our R&DC classes is pretty similar to that. Then you do alignment to the genome. Now, this step is a major differentiating factor between RNA-seq and ChIP-seq data analysis. So we had discussed two different types of tools that are used for these analysis. For RNA-seq, we had discussed star aligner. HiSat2 is also used, but for ChIP-seq, preferably bowtie is used because the context in both of these experiments is different. So based on the requirement, based on your sample, and based on what you wish to align it to, some protocols are standardized. But of course, you can adjust parameters and you can use these rules interchangeably, but that requires more expertise. So if you are somebody who is new to bioinformatic analysis of such high throughput sequencing data, it's advisable to go for standardized pipelines and standardized protocols to avoid any incorrect steps in your analysis and incorrect outputs. Then you get your, after alignment, you get your SAM file, you convert that to your BAM file using SAM tools, you filter it, you call for peaks. Peak calling simply meant that whichever region has gotten significantly enriched when you have pulled down using your antibody of interest, that will represent a peak. More number of reads are aligning to that region. Then you can get the bed file from that, you perform quality assessment, and you will get an idea as to what region has gotten enriched when you align it to the regular human genome, and you'll be able to figure out that there is X-fold enrichment over the input, which means that compared to input, when I am precipitating my protein of interest, then these XYZ fragments of DNA are getting enriched, which represents that these are regions where the protein is my protein of interest is binding to the genome. So then you can do motif discovery, you can visualize it, and scope is endless. So that is the overall flow. If there was any doubt at any step, please interrupt me and we can quickly brush up those doubts. Following that, when we come to the steps, then the first pre-processing steps are quite similar. You do an initial data pre-processing using two tools. One is FastQ, the other is Streamomatic. There are other tools also. There's MultiQC. You can use that as well. There is Trim Galore. I think there are multiple options. Again, I'm showing you the standard protocol that you will usually find for most studies. And the details of the tools that have been used by the authors when they make data publicly available analyzed data. If you want to know which tools have been used for that, that information also you'll get from geodata sets. So you can read about that as well, so that when you report your findings from their analyzed data, then you make sure there is no discrepancy between what they have done and what you have done. So yeah, the first tool, FastQC, is basically a quality check or quality control tool, which helps you to check a few parameters, which we have already discussed in a lot of detail. The sequence quality, the idea that on the flow cell, which was being used for sequencing, if there was any bias, where there was any tile bias, any particular region showed very low quality reads. What is the distribution of GC bases, the length of the sequence, whether there's any duplication or not, overrepresented sequences, the removal of adapters, all of this can be checked using FastQC. So after initial FastQC analysis, you perform trimming so that you can get rid of reads which are of low quality, which do not satisfy a certain threshold. If you remember, we had talked about sliding window strategy for analysis, where where the tool would be able to scan through the sequence within a window, calculate the score, and if the score does not meet a certain threshold, if the Phred score does not meet a certain threshold, then that region would get trimmed, versus if it meets that threshold, then it would be retained. So these kind of trimming strategies can be used. And finally, what you get is your raw read, which has been trimmed to meet basic parameters which are necessary for downstream tools to be able to do their job, and which does not carry any adapters, which does not carry any bias sequences, which gets rid of low quality reads. So all of that has already been adjusted. So the trim sequence is what you will use downstream for your subsequent analysis. Next, we come to the alignment step, which, as I mentioned already, was the um, key difference between RNA-seq and ChIP-seq data analysis. In context of ChIP-seq data analysis, the tool that we discussed was Bowtie or Bowtie 2, which is what is usually followed. So Bowtie 2 is an alignment tool that also follows a similar strategy to Star Aligner in the sense that it first fragments the genome and indexes it. If you remember, indexing simply means sort of uh, sort of classifying small segments of the genome so that it becomes easier to identify and easier to detect which region it is exactly aligning to. And POTI2 does gapped, local, and paired end alignment as well, and multiple processors can be used to make the tool more efficient. Its output, what you will usually get is in the .sam format, which is an alignment format, and then that can be converted into the BAM format, which is binary and much smaller, much easier to handle, and much more preferable by other tools. So, Bowtie 2 is often the first step in multiple pipelines for comparative genomics, including variant calling, ChIP-seq, RNA-seq, um, bisulfite sequencing, etc. So yeah, that's what it does. And as you can see, the output that you get from Bowtie 2 is the SAM file post alignment, which then can be converted into .bam files, which make it easier to study, interpret, and handle overall. So, how does it work? If you remember, it indexes like I already mentioned, and then it performs alignment. So, alignment is basically a process where it uses the human reference genome, which, if you remember, you can retrieve from multiple sources, but from the UCSC genome browser, for example, you can get HG38, which is the most recent available version of the complete human genome. So you get the EDGE38 sequence and you align your reads and your sequences that you have derived after immunoprecipitation. And then you check which regions of the human genome it is able to align to. So that shows you the percentage alignment. So it basically tells you where your read has come from. So this is what it approximately looks like. And then there are different types of alignment that Bowtie 2 can do. For example, by default, it does the end-to-end alignment where it looks for all of the characters to be matched to a certain region of the genome. And it is also called as an untrimmed or unclipped form of alignment. When a local option is specifically mentioned, then Bowtie 2 is able to perform local alignment, which comes next, and then it can trim or get rid of or clip some regions, which you can specify, like the trailing five bases or the leading five bases, all of that can also be adjusted. So this was an example of an end-to-end alignment, which I've already discussed. Is it not changing? Yeah. This is an example of the local alignment that I was mentioning. So in this case, you can see there are four characters that have been soft trimmed or have been omitted from the beginning and three characters from the end that have been omitted. So that local alignment score can be maximized to make the alignment more practically feasible. And higher scores, of course, indicate higher similarity. And you can refer to this manual for more details, like I already mentioned in the previous class. And this was the a recap of the flow that is to be followed. So first is quality control using FastQC and Trimomatic. Then the FastQ file can be used for alignment to the human reference genome using Bowtie2, which is going to give you a BAM format file, which can then be filtered and converted into from it is going to give you a SAM file which can be converted into the BAM file using SAM tools and SAM Bamba. Sorry, that throat. And then the BAM file can be used for peak calling, and the bed file that you get out of it can be visualized using IGB or integrated genome viewer or browser. Then comes the uh, peak calling part, which, as we already mentioned, is basically going to give you enriched, which is going to help you visualize which regions of the genome are enriched when immunoprecipitation is performed. So peak calling results usually are given in the bed format, and although ChIP-seq peaks do not give you strand information that which strand it has come from, but you can get an idea about which strand it has come from based on the histone marks around the TSS, because it will detect or it will dictate how much the chromatin has been locally opened up in that region. However, the accuracy of these tools is never 100%. And so a practical strategy is that you try to remove as much noise as possible, you remove subgroups, etc. All of this detail you don't need to get into. Basically, the idea is to identify those regions which are enriched. So that is indicative of the peak, which is representing higher or propensity of your protein to bind to that region within the genome. So if we look here on the right, you can see this is your sense strand and antisense strand chip enriched fragments that you can get, and then you align it to the reference genome, and the type of tags that you will get uh, corresponding to the sequence region is going to indicate which side there is enrichment, and from there you can detect where the protein enrichment has occurred. Again, don't worry, this is something the tool is able to do. If you want to get into more details about it, you would have to read about it, but basically the concept is what I'm trying to explain to you, that you want to know the region of the genome, and specifically why is the sense or the strandedness of the genome a factor here. That is because you want to eventually go for motive discovery. You want to know what is the nature in which the protein is able to access the DNA. For example, if this is double-stranded DNA, whether the protein sits like this or sits like this is going to make a lot of difference. It's going to dictate how the chromatin folds. It's going to dictate which is the sequence that gets recognized. It is going to dictate the orientation of the protein, and whether or not some other XYZ proteins can also be recruited on top of your protein of interest. So it changes a lot, but the way the tool does it is something that is going to require a little more depth of understanding. So we'll keep it out of our covering topics for today. But overall, the idea is that peak calling is a very important step. Now, this peak calling tool, as I previously mentioned, is MAX2, which stands for Model-Based Analysis of ChIP-seq, whose job is basically to evaluate the significance of enriched chip regions. And it was initially developed for detecting transcription factor binding sites, but it is also used for the other purposes. So for MAX2, the peak calling function, MAX2 call peak, it basically identifies regions where more number of sequencing reads are able to align compared to the background control. Broad and narrow peaks also make a lot of difference. It can identify narrow, sharp peaks, for example, transcription factors, or it can identify very wide, diffused peaks, for example, for histone proteins, which have less stringent binding. Again, the types of peaks are something that I had discussed previously. Then it can do model-based read shifting. It models the bimodal enrichment pattern to estimate fragment size, shifts reads towards the center for more precise identification of the TFDS or the transcription factor binding site. Then it can filter duplicate reads based on sequencing depth, which is necessary so that any false quantification is not done. Control normalization, where it uses control samples or input DNA to scale the background and accurately calculate fold enrichment to reduce noise. Paired and support is also supported, which can calculate fragment size, improving accuracy. So that's pretty much these are the characteristic abilities of MAX2, and you can read more about this using the link, which is going to give you an overall perspective of MAX2 use or the use of MAX2 for ChIP-seq data analysis. Finally, after your analysis is complete, if you wish to visualize your data, if you wish to represent it, two common tools are used. One is Integrated Genome Browser and Integrated Genome Viewer, IGV and IGB, both of which are accessible online. So two things need to be done. One, you need to have your bedgraph file or your .bed file, which is going to give you these representation of peaks. And apart from that, you need to have your bigwig file. So usually for publicly available already analyzed ChIP-seq data, you will find these big files which you can use directly for your analysis if you wish to align with the parameters that have already been standardized. But if you wish to change any of those, if you wish to adjust it based on your requirements, your hypothesis, you can do that as well. And then you can load all of these tracks along with the human genome, and then you can detect which conditions are going to affect the binding of your protein of interest to the expected regions. So this is what the overall view of an IGV workspace looks like. So first you have the input where there are very tiny or almost no peaks. Then you have all of your different ChIP-seq samples, and at the bottom, what you see is a representation of the human genome. It shows you the chromosome number, it shows you exons and introns, and it shows you the exact coordinates where it is able to bind. So a few things to keep in mind. First is that whenever a comparison is to be made, you must always make sure that all your tracks, they have the same scale along the y-axis, which is usually, if you see here, it's represented by a number from 0 to 20. 0 to 20, you can see that all of them have 0 to 20 shown. However, 2 or 3 of these peaks here are getting cut off, so it's advisable to try and adjust the scale such that all of the peaks are at least detected properly. So maybe 30 would have been probably 30 would have been more appropriate for the scaling. And then you can also do 2 types of comparisons. One is you can calculate the fold change, the log2 fold change, which compares input versus your IP sample, or you can also do a direct subtraction of both of these tracks. So track 1 minus track 2, which is also going to allow you to compare between different antibodies if they've been used for immunoprecipitation, so that there also a comparison can be made. Or you can compare IP minus input also, so that you can get a subtracted track difference as well. So that is going to give you an overall idea of the relative enrichment of your protein of interest in your regions of interest within the genome, which is going to align, hopefully going to align with your hypothesis. Now, after this analysis is done, the next step is to validate it. How do you validate it? Or why do you need to validate it? That is because any high throughput sequencing data is always carries the possibility of some false result or some error, because as you can understand, the scale of the experiment is quite immense. So it is prone to error, it is prone to sequencing errors, it is prone to alterations, it is prone to some false positive, a number of things can go wrong. So whatever you have found using a high throughput sequencing data, you must validate it in vitro using standard techniques. So commonly what is done is the ChIP qPCR, which we have already discussed. So for example, if you find that compared to the input sample, you find in your IP sample that there is enrichment at this particular location, then you can design primers, genome PCR or qPCR primers specific for this region, and then use that to check whether there is enrichment of your protein of interest after ChIP or not. So pretty much that is what happens here. You can also compare 2 individual files using BAM coverage or BAM compare. A number of possibilities exist. You can go on GitHub and you'll find so many beautiful ways of representing your data. But for initial validation, these crucial steps need to be followed. Then opportunities are endless, and you can opt for whatever works for your case. So that was a slightly lengthy recap of the previous class. If you have any doubts, please let me know, and then we'll move past it and we'll enter attack sequencing, the concepts of chromatin accessibility profiling using attack-seq. So let me know, then we can start this topic.
No, ma'am, everything was clear. Like
Okay.
it was a revision, so it was like
Yeah, you were saying anything?
no, no, ma'am.
Okay. Uh, okay. So first of all, before I begin the discussion, conceptually, what you need to understand is that what is the logic behind chromatin accessibility profiling? Why are we so bothered about it? And why is it not always called sequencing and sometimes called as profiling? So chromatin, um, if you remember our previous discussions, we had already like hinted at it a little bit. Chromatin is not as linear and as simple as we usually interpret it to be. It is complex. It bears different types of structures, and its 3D structure has a lot of influence on how accessible it is and how much of the information it is going to disclose. So access to the chromatin, orientation or structure of the chromatin, and the expression of chromatin are all kind of interlinked, and techniques that are used to study it have become extremely relevant in today's time. So if you look here, first of all, just a second. Yeah. So when we talk about chromatin accessibility, it refers to the compaction of the chromatin in 3D space or physically, and the complex which is formed by DNA and the associated proteins, which consist of histones, transcription factors, chromatin remodelers, chromatin modifying enzymes, etc. So, what do we mean by chromatin modifiers and chromatin remodelers? Remodelers are those which are going to open up or close chromatin or distort the structure or the architecture of chromatin, whereas chromatin modifying enzymes may either physically do so or they might add some sort of a substituent group onto the chromatin, changing its accessibility. Most commonly, it is methylation. So a methyl group may be added that is going to restrict the access of transcription factors and other proteins or the scaffolding of these proteins onto the genome. So those are chromatin modifiers, and chromatin remodelers are those which are going to physically remove histones and other bound proteins, making the chromatin accessible. And the group of proteins which fall into these families are very different. So it's sometimes interchangeably used, but it's not correct. You should know the different classes of proteins. For example, chromatin remodelers can include switch SNF proteins. Most commonly, these are ATPase bearing proteins. Why? Because a lot of energy is required and has been invested by the cell to wrap up the chromatin within a very compact space. So the assembly of the histone proteins, the binding of DNA around histones, the formation of nucleosomes, the packing of multiple nucleosomes, forming the solenoidal structure, all of those things, they are very lengthy, very energy consuming processes. So disrupting that energy investment and opening up the chromatin is something that is going to need additional energy to be invested by the cell. And that is usually contributed by these ATPase containing proteins. Why? Because they are going to make sure that chromatin unpacking or chromatin remodeling is not something that can be passively done by the cell, can be tightly regulated by the cell, and C is done under conditions where the cell has sufficient energy access or sufficient resources available to it so that it can dedicate energy to a process which is not survival essential. So these chromatin remodelers play a very crucial job in determining whether or not the cell can afford to go for this unpacking and remodeling at that point of time. So how do you study it? So traditionally it is probed by assays which involve nuclease digestion. We'll discuss some of it today and some of it has already been discussed. So nucleases or restriction enzymes can be used for digestion. Typically, few regions of the genome are explored at a time. But when you go for a high throughput approach, which involves NGS, then chromatin investigation can be done in a more comprehensive manner. You can look at the entire genome at one go in a snapshot, and that you can use for your study. So one of the first genome-wide profiling experiments of accessible chromatin was published in 2008 by sequencing genomic DNA fragments following digestion by DNase 1, which preferentially introduces double-stranded breaks in accessible chromatin, which is referred to as DNase-seq, or basically you digest with DNA and then perform sequencing. So what exactly is done in that? In DNA-seq, first, we'll discuss this also, the difference between these techniques and ATAC-seq in the upcoming slides. But in DNA-seq, first, you isolate nuclei, you permeabilize it using a mild detergent like 0.1% right next 100, such that now the DNA is DNA enzyme is able to enter the nucleus and is able to access DNA and then make cuts. This is another very crucial step we sometimes don't shed a light on, which is that accessibility to chromatin also has to be provided physically by disrupting not just the cell membrane barrier, but also the nuclear membrane barrier, because that is also not, it's not porous. It doesn't allow the entry of any random protein, any random enzyme inside the nucleus. So permeability needs to be sometimes induced, uh, not only for experiments like this, but anytime you want to study anything in the nucleus, that needs to be done. Even for something as simple as immunofluorescence, for example, if you want to stain something which is a protein that is localized only within the nucleus, then it's advisable that you permeabilize the nuclear membrane. You use some sort of a detergent like Triton X100 or Tween 20 that is going to like make teeny tiny pores on the nuclear membrane and allow the antibody to enter. So similar to that, for DNA's enzyme to be able to do its job, you need to allow accessibility, which is why you need to make the nucleus permeable. Now, once that is done, the small DNA fragments, which are like 50 to 100 base pairs, they can be purified. They can be purified and size selected for downstream library construction and sequencing. Now, DNase 1 is an enzyme. It's a digestive enzyme. So the amount of enzyme can influence the efficiency of digestion, and also by virtue of the efficiency of digestion and efficiency of the enzyme, it can also affect the data quality. So what you need to do, you sometimes might need to titrate it. You might need to standardize how much DNase 1 should be added so that digestion A does not A does not go wrong and B does not become inefficient, in the sense that incomplete digestion is taking place, and as a result you are getting false results that you are interpreting. So, in addition to fresh cells, DNA-seq has also been utilized for FFPE or formalin fixed paraffin embedded samples. So, even if you don't have a fresh sample, if you have a patient sample that has been fixed into blocks, into paraffin blocks, and you wish to check your hypothesis in patient samples, then you can use these as well. So, this is what it schematically looks like. You have your DNA. Sorry. You have your DNA, which is which is getting accessed by DNase 1, and then DNase 1 is able to make these cuts in these regions. These little scissors that you can see, they sort of represent where DNase 1 is able to cut DNA, and you're going to get reads from this uncut region, which was bound by your protein of interest, and that is what comes up as these peaks, representing the coverage of reads and representing that these are DNA stretches that are bound by protein and hence protected from DNA's digestion, suggesting that these are protein bound regions. So that's what we see. And compared to this strategy, how is attack-seq different? Just give me a moment, I'll do that. Okay, sorry for the interruption. Yeah, so compared to compared to DNA-seq, attack-seq shows you a different strategy completely. So please don't confuse these. I'll show a comparison towards the end about the stark differences between these techniques. But for now, let's keep our focus on attack-seq. So attack sequencing is a strategy that involves a specific type of enzyme, which is also a point of commonality with DNA-seq because there also you had a DNase. But in this case, the enzyme is completely different, very unique, and it is something which you might not have come across before. But the enzyme is a transposase. If you have studied transposons before, then you would be familiar with it. If not, please let me know. I'll give a quick recap of the same as well. So transposase, not the normal one, but it is a genetically engineered transposase. This is a hyperactive form of the enzyme, which is able to do something very unique, a phenomenon known as stagmentation. So what is that? We'll learn about it. So um, yeah, so it is an alternative strategy to investigate accessible chromatin profiles. This genetically engineered hyperactive DNA transposase, which is known as the TN5 transposase, it is preloaded with monovalent mosaic end adapters that can do two things at one go. What does it do? One, it cuts, which is the job that you expect from any digestive enzyme. When I say digestive enzyme, I mean a restriction digestion sort of an enzyme which is able to cut DNA. And what it can do simultaneously, which is what is the unique thing that TN5 transposase can do, is that it tags the accessible region of the chromatin or the nucleosome depleted region of the chromatin. This tag is what gives it the edge above all the established techniques, and the tag is what you will use eventually to study the region which is now an accessible form of the DNA. So if you look at this, just brief overview of what the technique looks like. So if you have this similar sort of DNA, if you remember this figure and the next, if you have a similar sort of region, what you would expect is that when you use the TN5 enzyme, the transposase enzyme, then what it is able to do is that it can cut DNA in this region and it can also tag the same using some specific type of adapters. Again, these are mosaic end adapters that you can use for your subsequent detection, and the reads that you get in this case will also align to that stretch of DNA. The output of both these experiments is the same, both of DNA-seq and of attack-seq, that you get those regions which are protected by DNA, which are protected by protein, and that DNA can be sequenced and that DNA can be analyzed. So what are the steps that are usually to be followed in context of attack sequencing? First, you of course have to isolate the nuclei. You preferably do not want the other regions of the cell. Why? Because mitochondria also carry some DNA, or there is some other contamination that can influence that can influence your results, your experimental results, then you should avoid those. So nuclei need to be isolated from the cell. You need to ensure that chromatin structure is maintained and any associated DNA binding proteins, including nucleosomes and transcription factors, they're all intact. So you cannot be very harsh in this method. You cannot be very careless in this method because here the 3D structure of chromatin also matters and you do not want to disrupt the same. Next, what happens is that the chromatin is exposed to the TN5 transposase, and then that acts as a homodimer where it does two things. First, it cuts the chromatin or it fragments the chromatin, and second, it introduces these sequences which contain PCR handles from where PCR can be performed. So, what is the relevance of that? It helps us to perform downstream amplification with I5P5 or I7P7 ATAC-seq adapters. You don't need to know the specifications of these. These are just types of ATAC-seq adapters. And I will explain the simplified steps in the next slide. And so only fragments that have these two types of adapters, adapter A and adapter B, let's just call them that for sake of simplicity. So fragments that have adapter A on one end and adapter B on one end will be amplified and subsequently sequenced. After sequence analysis of the library fragments, genomic regions which have been enriched for many transposition events are designated as peaks of chromatin accessibility or the ataxic peaks. So let's look at this image. So first you have your intact chromatin containing um, nucleus from where the chromatin has been isolated, and you want to conserve the 3D structure like I had already mentioned. Sorry. Next, you expose the cells to TN5 transposase. So transposase, as you can see, these green colored enzymes, it is going to bind to those regions of the chromatin which are accessible. It is going to cut through it. And on top of it, because it functions like a homodimer, so it has these blue and orange colored adapters, which indicate two types of adapters that need to be bound to the sequence where it is cutting. So next what you do is you isolate the transposed fragments and you amplify. And these are transcription factors in red and orange. And TN5 transposase. Okay. Next you amplify these regions. You sequence it and detect which regions they correspond to compared to the genome. So let me go to this next slide. So this is what the enzyme's job kind of looks like. So you have your region that is under investigation, and you have your TN5 transposase bound to bound to two regions. Now, after transposition has been done, what does the DNA fragment look like? So it has this TN5 induced nick, and you can see on one end there is adapter A, on the other end there is adapter B, which is necessary for PCR to proceed. Then initial extension is done at 72 degrees centigrade. The amplification and addition of barcodes and all the adapter components, etc., is all completed. Finally, the ataxic library is purified, and then sites which are accessible are then defined by the insertion of this TN5, this red and this blue. As you can see, these are the primers which have been um, introduced by the transposase. So those regions you can then study. Now, after TN5 insertion of the PCR handles, the nick, which is left behind by transposase, can be filled in during the initial 72-degree extension in the first step of the PCR. Then the fragments can be barcoded, sent for sequencing, and then you can trace back where exactly it came from within the genome. Now, what is the output that you get in this case? Sorry for the disturbance. There's some issue. Let's switch off my video. Yeah. So this is a representation of what hypothetical ataxic data might look like. Just a minute. Yeah. So what that data might look like. So for example, if you look at two different cell types, X and Y, they both have ataxic peaks at the promoter of gene A. So this is your gene A, and you can see peaks which are shown in purple and green respectively for types Y and X. And they're both present for gene A. However, when you look at cell type X, that has a specific enhancer which is upstream of the DSS data, which is here shown in green, and that is actually controlling the increased expression of gene A. So while there is enrichment of the ataxic peaks at the promoter of gene X, upstream enhancer shows differences between cell type X and cell type Y. So in the ataxic data, this would appear as a cell type specific ataxic peak, which would be differentially accessible between different cell types. In this toy example, what you see is that when there is differential accessibility at the enhancer by transcription factor B, then that is enhancing gene expression as well. So ataxic data can help us understand how transcription factor binding motif is associated with differential peak enrichment. And here it is depicted as GATAA, which is the motif for transcription factor B, and which is why in cell type X you see increased expression. Now, if we talk about the steps in taxic data analysis, or rather taxic process, then first, this is what you expect the difference in chromatin to look like. You have your chromosome that has both types of DNA, closed as well as open chromatin, depending upon which region is accessible at that point of time within the cell. And second, you have a hyperactive TN5 transposase that has two different adapters shown here as adapter 1 and 2 in red and blue. Now, when you start a taxi, how do you prepare your sample? First step, you have to harvest your cells, count them, lyse them, and finally obtain pure nuclei. And it is extremely crucial to maintain single cell suspension. You do not want any clumping, as that can potentially skew your results. Then the cells are lysed by a lysis buffer that releases the nuclei while keeping the chromatin intact. Then you can centrifuge, wash excess buffers, and finally have only your cell lysate. Second is probably the most crucial step, that is transposition. So what does it do? It does two things. First, it fragments your DNA or chops up your DNA, and second, it tags it with some unique adapters using an enzyme which is also genetically engineered, the TN5 transposase enzyme, and you create your taxic library. So this leaves the chromatin into smaller fragments, targets open chromatin regions, and then you can remove excess transposase and contaminants and proceed further. So this is what you see on the right. You have your TN5 transposase shown here in purple bind to open chromatin regions, and fragmentation and tagging or tagmentation by the TN5 transposase is such that it gives you these deep cuts or cleavage that is then attached to or is tagged by some sort of an adapter, which is why it is called as a tagmented DNA fragment. Then you have your amplification step, whereby your purified DNA fragments would have to undergo PCR. You increase more and more DNA that is going to allow you to perform sequencing. The final DNA library can then be checked for using gel electrophoresis or fluorescence-based detection methods, and you should have more and more of your desired DNA. Then comes the sequencing step where ataxic libraries are loaded into a sequencing machine. Most commonly, what is used are Illumina platforms. Paired end sequencing is also preferable. You'll have more data confidence and more information can also be obtained. Single end sequencing, while often used, is usually less informative and less reliable as well. So at the top, what you see is the example of just a schematic example of some data. So you have your peaks shown here in KB. You have sequencing peaks, which are corresponding to open chromatin. And this is what you kind of can see is showing you enrichment. Ataxic data analysis basically consists of four key steps. First is pre-analysis, core analysis, advanced analysis, and multi-omic integration. So what do we do under that? First, you do QC. Again, initial steps are the same. You check the sequencing data quality for errors and unwanted DNA sequences or adapter sequences. It uses fast QC for this purpose. Then GC content And sorry, what got lost. Yeah, so pre-alignment uses fast QC to check the quality of base, the sequence, adaptive contamination, um, quality of bases, repeated sequences, or overrepresented sequences, etc. Trimomatic is then used to remove low quality reads and adapter sequences, again, very similar to what we have previously discussed. Then comes your core analysis. What does that entail? That entails peak calling, which allows you to identify open chromatin regions. Most peak callers are adopted from ChIP-seq or DNA-seq methods, and there's only one peak caller that is specifically developed for ATAC-seq, that is called as the HMM RETAC, which is the hidden Markov model R for ATAC-seq. Then comes advanced analysis, which identifies the four key patterns in the data. What are the patterns that we're talking about? A, there has to be peak, there has to be motif, footprint, and the nucleosome. So you can annotate exactly which peak, you can detect for any expected motifs of transcription, um, whether there are any transcription factor footprints, or to study the positioning of nucleosomes. Lastly, you can combine it with multi-omics data integration to fully understand gene regulation. And multi-omic integration will help you compare ATAC-seq data with ChIP-seq, confirm binding sites, confirm using RNA-seq data as well, and then using reconstructing regulatory networks. So if there were any doubts in this part, please let me know and then we can proceed.
No, ma'am.
Okay. Now, what do we use it for? Okay, why at all did we get into ATAC-seq? Why? Because DNA-seq and all the previous methods that we have talked about, they need a much larger sample number. ATAC-seq requires very few cells, which makes it quite useful for more rare sort of samples. For example, if somebody is using, say, for example, patient data, or is analyzing patient tissue, or is using some specific blood cell type, or is using stem cells, for example, all of that makes sense. Utilizing small sample number for those strategies, it does make sense. Then it has a much simpler library preparation method, making it easy and quick in terms of completion. You do not need prior knowledge of epigenetic mechanisms. You can get very like you can get very confident results from very little sample, and very reliable analysis can also be performed because it has high sensitivity. It can accurately detect open chromatin and regulatory elements both in the bulk method and the single cell sequencing attack sequencing method. It provides quick results, which is suitable for large scale applications like clinical research or drug development, etc. And finally, it can be used to understand diseases that are linked to epigenetic challenges, including cancer, autoimmune diseases, and neurodegenerative disorders. So applications are quite immense. We'll come to the application also. Then come the limitations of ATAC-seq, which only captures chromatin accessibility. It does not give you any information about DNA methylation, histone modifications, and chromosome interactions. TN5 transposase, which is used in ATAC-seq, can also lead to bias in adapter ligation. Sometimes it can insert the same adapter. If you remember the I5 and I7 that might be inserted, um, like the same adapter may be inserted to both ends of a DNA fragment, and that would not be used. These fragments cannot be used for sequencing, uh, which are obviously going to lose out, which is why you're obviously going to lose out on a lot of data just because incorrect tagging or incorrect adapter ligation has been done. And that is also going to skew the coverage of your genome. It's going to lead to an uneven coverage of the genome. PCR amplification step can create challenges when you're dealing with very large DNA fragments. Computational challenges also persist. Researchers often rely on tools which have been designed for ChIP-seq and DNA's seek to analyze ATAC-seq data. Currently, not a lot of unique tools are there, but of course, they are up and coming, and within a few years, we'll have more clarity regarding the same. So, what are the applications? Firstly, of course, it is to identify open chromatin regions, which will let you study transcriptionally active sites or transcriptionally active stretches within the genome. Secondly, you would get an idea to connect not just this data, but connect it with RNA-seq data, connect it with ChIP-seq data to altogether have a comprehensive validation of your expected hypothesis. So it is used to study gene regulation and transcription factor binding in different biological processes. So that includes many complex diseases, immune responses, and also cancer. Thirdly, it is also used for mapping of nucleosomes, which helps us understand how chromatin structure can influence the activity of the genome. It can be used overall for cancer epigenetics as well. And it is also used in disease research to map disease-specific chromatin alterations, identify those regulatory elements that are associated with diseases, and discover potential therapies that target chromatin accessibility. It also has roles in comparative genomics and evolutionary studies as well. But overall, this is the idea. And if you look at the right, the diagram shows you sort of an immediate comparison between MNAs, DNAs, and attack sequencing strategies. So in MNAs, as you can see here shown in red, digestion is going to give you those nucleosome bound DNA stretches, which come up as individual peaks for each of the nucleosomes. As you can see here, these tiny red peaks are visible. Second is your DNA's seq, which is going to chop up everything else except for the completely open chromatin. And that is by virtue of the way in which these enzymes work, or how they approach or access the genome. And as a consequence of DNA seq, you would get the enrichment at only the central region. Then you have a tag seq as well that is going to represent the regions which have been tagged by the TN5 transposase. And these regions also come up as these individual hits, which are shown here in yellow. And the sequencing of this particular region, which has, of course, remained inaccessible to the transposase enzyme, has been by virtue of a protein binding at that site. And that shows that there is a concordance between MNAs, between DNAs and attack sequencing. However, the sensitivity and accuracy is much, much higher in, um, in attacks. Yeah. The other very commonly used strategy for chromatin profiling. So we have discussed three today. I think I only have like a few slides left. Yeah, so we'll wrap up the class pretty soon. But apart from that, there are a few more techniques, like there's high C as well and high chip as well. So I'll briefly discuss that in the next class. So before we move to that, first we understand MNA seq. So while you've seen the previous image, um, MNAs, the micrococcal nuclease, as we already know it, um, is also used in a similar context. So nucleosome positioning and occupancy in the genome plays a very important role in accessibility to the chromatin. So MNase is an ectoexonuclease that is able to cleave DNA regions that are not nucleosome protected, and that leaves the nucleosome core particles undigested, which can then be purified, ligated to adapters, PCR amplified, and then can be sequenced using MNA-seq. So it is therefore an orthogonal assay compared with DNA-seq and ATAC-seq. Why? Because it measures nucleosome occupied regions and is most widely used to map nucleosome positions throughout the genome. A recently developed quantitative protocol. Okay. a recently developed quantitative protocol for MNase-seq that involves subjecting aliquots of the sample to multiple MNase digestion levels or the different quantities of MNase that allows a very clear distinction of nucleosomal positions and occupancy from higher order chromatin profiles that can be summarized as well. So what does it do in MNase-seq? It uses either 10,000 to 100,000 cells that are either fresh tissue samples or they can be paraffin formaldehyde fixed cells that can be used for light reconstruction. Then MNase digestion usually will result in a nucleosomal ladder. Again, I think in our initial discussion on the epigenetics studies, I think in week 4 or 5, somewhere around that time, we had discussed how MNase exactly works. Yeah, so that gives you a nucleosomal ladder. So you will have mononucleosome, which will obviously migrate fastest through a agarose shell, dinucleosome, a little bit of retardation, trinucleosome, so on and so forth. So based on how much MNase is actually provided, you will get this gradient. And the optimal range of digestion usually leads to 70 to 80% of mononucleosomes, 20 to 30% higher order nucleosomal ladders. So every region that is nucleosome protected, uh, nucleosome protected can be sequenced. So yeah, that's what you see. So overall, if you look at the different strategies which are present for studying epigenetic modifications of the chromatin, then there are three common strategies. You have ATAC-seq, you have ChIP-seq, and you have MNase-seq. So ATAC-seq shows you the possibility where TN5 transposase binds to open chromatin. You get your tagmented DNA sequences, which can then be sequenced. Second is your ChIP-seq, which we have discussed extensively. So that involves your DNA binding protein, using antibodies against your DNA binding protein, and fragmenting, sonicating any other such unbound region, and then detecting it. Third is your MNase-seq. So MNase uh, digests, and what you get are protected and bound DNA, which can then be isolated, and then protein binding sites can be detected. So that was pretty much what I wanted to show for today. If you have any doubts, you can let me know, and then I'll discuss my plan and I'll see if I can show you some other images or not. But yeah, first, any doubts, please go ahead with that first.
Ma'am,
You
I
all
am a little bit confused between the ataxia and the last one. What is the major difference? Like you told, but I was like,
Okay, so the first difference lies in the enzyme which is used. Transposase is, in this case, is a genetically modified transposase. So apart from just cutting DNA, what it is also doing is it is tagging it. So there is an adapter which is ligated at the site where it is cleaving. And as a result, when, if you remember our initial sequencing, like the discussion, overall workflow of sequencing, if you remember the analysis part when it is to be done, the adapter helps in amplification. So here. So if you look here a little closely, you'll see that apart from the nucleosomes,
ma'am, are
the
you
rest of the
sharing your
region.
screen?
Oh, I'm so sorry about it. I'll just share it again. Yeah. Wait, let me go back to that slide. It'll make it easier, maybe. Yeah. So if you look here, uh, the enzyme by itself at the top, if you look here, the enzyme is shown here as a dimer, right? So there are two units, each of which are um, they carry an adapter on themselves. So what happens during this process is that when transposase by itself is working, what is it able to do? It is able to cut the DNA. Like you can see here, there is a cut by these two enzymes, and simultaneously, what it is able to do is add on the adapter. The cut is also known as a nick in this case. So then it is able to insert this adapter, which you can see in red or in blue on either side. Let us look at the legend. Maybe that'll make it easier for you to understand. If you look at the legend here, the final sequence that you will get, that consists of this black region, which is your original ataxic fragment. This is the region that is that is staying put as it is. Next, you have this red region, which is your I5 unique TN5 overhang. I5 is a sort of an adapter, which is or I5 is a unique sequence, let's put it that way, where it is after TN5 transposase is cutting, it is leaving an overhang. Next is this yellow region, which is a barcode. That is your adapter. That is going to help you eventually align your read to the genome. It is going to help you figure out where it has come from, which cell it has come from, and trace back the origin of that cell. Next is your think of it like a QR code. Let's put it that way. A barcode or a QR code. It's basically to trace the origin, cell of origin. Next comes your flow cell adapter, which is shown here in red. Now, the flow cell adapter will, of course, depend upon which platform your sequencing is being done on. All of that is taken care of by the company. Then you have this homology region, TN5 common homology region. Why? Because TN5 has to find the region which it can access, and then it can do its job. It can work as a transposase at that site. Then on the other end, you have this blue fragment, which is TN5, the other adapter, and the I7 barcode for the two, the second adapter, and P7 flow cell adapter is another flow cell adapter corresponding to where it is going to get sequenced. So the key difference between ATAC-seq and MNA-seq is A, the enzyme which is going to do its job. It is very sensitive. It is not going to miss out on any regions. Secondly, it is tagging your fragments. Basically, you are putting an address on your fragment so that whatever information you get out of your sequencing experiment, you will be able to trace it back to the cell of origin, and you can rely on the accuracy of the method as well. And yeah, mainly these two, the enzyme, the type of data, and the type of fragment which is generated, that is completely different between these two processes. So you need a little bit of background about transposition for it to work. I hope you've studied about it. So this is a transposase dependent strategy. There are others also, like there's a cut and run strategy. There's a MNAs coupled with cut and run as well. We'll discuss all of that in the next class. After high C, I'll come to those strategies. So that'll pretty much complete our chromatin profiling concept. There's also 3C. There's also that technique. These are all basically techniques which are going to give you similar outputs. It is going to tell you which region of the chromatin is accessible and which of it is not. But the logic of using these or deciding which strategy you need to use depends upon how accurate it is, how much information you can get from one experiment, and thirdly, what is your hypothesis that you wish to address. So MNAs and Ataxic is going to give you kind of similar outputs. If you remember this, where is it? Yeah, this figure, not MNA, sorry. Yeah, MNAs and ATAC-seq is going to give you similar outputs in context of nucleosome um, bound regions. And the commonality between DNAs and ATAC-seq is the fact that it is going to give you completely opened chromatin ka information also. But if you see, ATAC-seq gives you both these types of information. The difference being that it is going to tag those regions so you can trace back where it has come from. So it is going to give you more information in one single experiment and also make it much more reliable. So overall, these are the differences. But make sure if you are ever like spotting the difference, you have to make sure you mention the starting material difference as well. Uh, these DNAs, MNAs dependent reactions, they require a lot of material. Ataxic does not require that much nucleus or that much starting material. Secondly, the enzymes, MNAs and DNAs, beyond a point can have non-specificity, can have off-target effects because they are just physiological enzymes that we have purified from some source or another. DNAs is purified from some organism, MNAs from some other. So they are not genetically modified for high fidelity, whereas for ATAC-seq, the TN5 transposase is uh, engineered for hyperactivity, but also with more accuracy. So that gives it an edge above others. So I hope I could clarify your doubt. I said a lot of things, but I hope you understood.
Yes, ma'am. Now it is clear.
Okay. Actually, I was looking for a video if that was possible. I couldn't get it, but I'll see if I can get a TN5 transposase, the mechanism that it follows in more detail, because I think that'll help better. But yeah, pretty much that is what it does. Uh, we'll discuss the remaining strategies. I think I have the syllabus here. Yeah. So in 22, we are going to discuss high C, and I'll just briefly introduce 3C as well. Um, these are all relatively newer techniques, but if you go to really high impact journal papers, you'll see that people are talking about it. If the objective is to study what does the enhancer do, what like to go beyond just gene expression and see how chromatin is controlling gene expression. All of these techniques are like superb and very expensive, but be very informative and going to give you so much information. So we'll discuss that. And these topics are quite simple. So I'll just wrap it up in one class. So in the next 2 or 3 classes, we'll finish these remaining topics. And in the final class, we'll have our discussion and a quick, it'll be like a questionnaire. It's not really also going to be that formal. But yeah, it'll be a summary of the entire course. So anything you wish to discuss in the upcoming in the last class or in the coming classes, please let me know. And if there's anything that has not been clear throughout the course, then now is the time because we'll wrap up very soon and then it's not going to be possible. But please let me know then as well. Okay. So if there's anything you want to discuss today, please let me know, else I'll end the class for now. Okay, if not, then thank you for joining. I kept today's class brief because it's a complicated technique, but I think next class might be a little more loaded because they are more, these are unfamiliar techniques, but it's good to be updated with literature because people are doing these left, right, and center. Any well-funded lab is going to do this. So if you want to go somewhere, it's good to at least have an idea about the technique and then you can keep learning it throughout your journey. That's going to be endless. So, okay. Anyway, thank you for joining. We'll continue our discussion on Friday, which is, I think, 27th. Yeah, 27th from 6:30 or 7 p.m. You preferably at 6:30 only, but I'll drop an email to confirm the time once more. So, okay. Thank you for joining. Have a good night.
Thank you, ma'am.